Formation of germinal layer. There are three germinal layers: ectoderm, endoderm, and mesoderm. All these three layers are derived from the embryo proper, and it will give rise to all the tissue of the body, all the tissue of the human body, as well as the supportive structures to the developing fetus with help in acquire the nutrition. So in this lecture, we are going to learn about the formation of these three germinal layers. As you can see in this picture, after fertilization, zygote is formed. Zygote is a single-celled structure. So for the further development, there must be division. Then the process of division is called cleavage. Cleavage is a process of mitotic division, occurs in the zygote, and it gives rise to number of cells like 2 cell, 3 cell, 4 cell, and so on. So this process is called division of cells is called cleavage. This process of cleavage have three stages. One is stage of compaction, morula stage, and blastula stage. Stage of compaction is the 8 cell stage, morula is the 16 cell stage, and blastula is the 32 to 64 cell stage. So as you can see, this fertilized ovum is divided into 2 cell, 3 cell, and so on structure. And finally, we got the morula stage. This is morula stage. This morula stage, you can see there is a mass of cells. This is just the mass of cells, divided cells, that is surrounded by a layer known as zona pellucida. And all these changes inside the fertilized ovum occur when it travels through the fallopian tube to the uterine cavity. We will discuss it later about the zona pellucida. It is a very important covering, and it will remove just before the implantation. It got removed just before the implantation inside the uterine cavity, because as you can see in this next picture, this inner cell mass before this was just a hub of cells, that is the mass of cells. Mass of cells. Now this mass of cell differentiate itself into two type of mass. One is known as outer cell mass and one is the inner cell mass, or we can say outer cell mass and inner cell mass, that is surrounded by the zona pellucida. You can see the persisting zona pellucida. And at this stage, after the morula stage, we have discussed the blastula is formed. So now this arrangement of cells is known as blastula. In next stage, you can see the outer cell mass. Inside this, a cavity is formed and the fluid accumulates. Due to the force of this fluid, the inner cell mass pushed towards the one side of the outer cell mass. The place where the outer cell mass to the outer cell mass, inner cell mass is attached is known as polar trophoblast, and rest of the trophoblast is known as mural trophoblast. So in the formation of germinal layer, you can see there is a single zygote, then 2 cell, 3 cell, 4 cell stage. This process is known as cleavage. Then we got the 16 cell stage, that is morula, just a mass of cells, and after that we got the blastocyst, in which the inner just the cell differentiate itself into the outer cell mass and inner cell mass. Then there is a formation of cavity, or we also called it blastocyst cavity, and the fluid push the inner cell mass towards the one side, and it divide the trophoblast into two parts, polar trophoblast and mural trophoblast. Now here is a very important thing to understand. As we discussed in the first slide, from the all three germinal layer, whole of the tissue is developed, plus the supportive structure developed. So all the supportive structure are developed from this trophoblast. It will not give rise to any tissue of the body, any embryonic tissue. Okay? And this embryo proper, this is called embryonic tissue, and it will give rise to the these all three germinal layer, that is ectoderm, endoderm, and mesoderm. Okay, all these three germ layer are derived from the inner cell mass, and all the supportive tissue, supportive structure, we can say, like umbilical cord, placenta, fetal membranes, connecting stalk, all these is derived from this outer trophoblast, and all three germinal layer are derived from the inner cell mass. In this picture, you can see for in the further development, when outer cell mass and inner cell mass differentiate itself, then this inner cell mass, few of the cell from the inner cell mass come downward and lie to in the this position. This is the beginning of formation of first stage is formation of hypoblast. This is known as hypoblast, which is going to be later on endoderm. This is the endoderm in future. Rest of the cells of inner cell mass become columnar. You can see in this picture. This is known as epiblast, which is later going to be the ectoderm. In the next stage is the formation of epiblast. In the third stage, you can see there is a formation of cavity. These yellow color cells are derived from the trophoblast. These amniogenic cells are derived from the trophoblast. It means it is again not going to give rise any the tissue, but the supportive structure only. So you can see next stage is the formation of amniotic cavity. Its floor is formed by the epiblast and roof is formed by the amniogenic cells, and amniogenic cells are derived from the trophoblast. In next stage, that is the fourth stage, you can see the cells of the hypoblast lines itself in a form of a cavity and form a membrane initially, that is also known as Hisner's membrane, or you can see the name of cavity is primary yolk sac. So fourth stage is the formation of yolk sac, primary yolk sac. So you can learn quick revision again. So after the outer cell mass and inner cell mass differentiate in itself, then inner cell mass further undergo differentiation. There is a formation of hypoblast, then there is a formation of epiblast, then there is formation of cavities of for the each layer. There is amniotic cavity and there is a formation of primary yolk sac. So at this stage, the we can say there is a bilaminar disc formation. The disc name is the bilaminar, as we have two layers only, epiblast and hypoblast, which is going to be future ectoderm and endoderm. So in this picture, you can see next stage is the formation of this pink color structure, that is the nothing but the extra embryonic mesoderm. This extra embryonic mesoderm is derived from the trophoblast. Trophoblast undergo division and give rise to a mass of cells that adhere together to form the extra embryonic mesoderm. So it means again extra embryonic mesoderm again going to give rise the supportive structure, but not all three germ layers. So at this stage, the germinal disc is of two layers. We have two cavities, and we have an outer cell mass, that is trophoblast, and the mass of a tissue that is known as extraembryonic mesoderm. In the next stage, you can see in the extraembryonic mesoderm, a cavity is formed. This is the cavity of extraembryonic mesoderm, and you can see this cavity of it is present in a small firstly, this is a small cavities, and later on it adhere to become a large cavity. And as you can see, this extraembryonic cavity, extraembryonic mesoderm cavity, not divide the whole of the extraembryonic mesoderm. It will leave a area which is going to be the future connecting stalk, which is again a supportive structure, and you can see it is again derived from the extraembryonic mesoderm. As this cavity divide the the extraembryonic mesoderm is divided into two parts. One part that is close to the trophoblast is known as somatopleuric extraembryonic mesoderm, and one part that is close to the embryo is known as splanchnopluric extraembryonic mesoderm. This is the one event, and second, one more important event occur. There is a formation of two important fetal membrane, that is amnion and chorion. These membranes are again supportive structure, and as you can see in this picture, it is again derived from the outer cell mass tissue. So at this stage, we can say chorion equal to trophoblast plus somatopleuric extraembryonic mesoderm, and amnion equal to these the amniogenic cells. It is made up of amniogenic cells plus splanchnopluric extraembryonic mesoderm. So you can see all these structures are derived from that outer cell mass or trophoblast, we can say, and it has nothing to deal with this inner cell mass, or which is later going to be form the three germ layer. So at this stage, the important phenomena after the formation of outer cell mass, cell mass, then there is a formation of epiblast, hypoblast, then there is a formation of epiblast, and there is a formation of amniotic cavity, then primary yolk sac. Next, there is a formation of extraembryonic mesoderm, then there is a formation of extraembryonic coelom, which not divide whole of the extraembryonic mesoderm, and it will leave a part that is going to be the future connecting stalk, and it will split the extraembryonic mesoderm into two parts, splanchnopluric and the somatopleuric mesoderm, and it also give rise to the two important fetal membranes. And so at this stage, we just take the two layer structure, or we can say bilaminar disc. In this above is, we can say, ectoderm, and this is the endoderm. As you can see, at this stage we can't decide which part is going to be the head and which part is going to be the tail. So there is a important structure that decides the which part of embryonic disc is going to be tail and which part is going to be head. So for this, you can see in this picture, there is a formation of procaudal plate. That important structure is the procaudal plate, and there is a overgrowth of the cells in the hypoblast cells, or we can say endodermal cells, outbulging of cells, that is known as procaudal plate. As this procaudal plate is formed, we can say this part is going to be the head and tail. And in this picture, you can see this is the head and this part is going to be the tail. After the formation of procaudal plate, in the caudal region of the epiblast, thus again a changes of cells or growth of cells occur, outbulging of cells, that is known as primitive streak. That is known as primitive streak. As you can see, the next stage is the formation of primitive streak, and here is important event, the procaudal plate which going to decide the head and tail part, and next is the formation of primitive streak. This primitive streak begin the formation of the third layer, the very important layer, that is known as intraembryonic mesoderm. Always remember, before this we learn about the extraembryonic mesoderm, so we have to name it the intraembryonic mesoderm, that is the actual mesoderm. You can see the cells from the primitive streak outgrow and push itself in between the epiblast and hypoblast, or we can say ectoderm and endoderm. And in this way, the third layer, that is intraembryonic mesoderm, is formed. So important structure for the formation of intraembryonic mesoderm, or we can say the intraembryonic mesoderm is also derived from the ectoderm, because the primitive streak form inside the ectoderm. So for the further detail we will study in next